Hi everyone, welcome back. This is lecture 6B of Useful Genetics, and we're going to extend the previous lecture's discussion of DNA fingerprinting by talking specifically about the genetic markers that are used in this analysis. We'll talk about what VNTR alleles are and how and why VNTR mutations arise so often. And then we'll describe the set of loci that was developed for forensic work. So, as we said, each VNTR marker, each place in the genome that's a VNTR marker, has many different alleles. They're highly polymorphic. And this situation exists because of the nature of VNTR loci. VNTR stands for variable number tandem repeat loci. Loci are places on the genome. We'll unpack this backwards. They're repeats. They're short sequences present in multiple copies. As you see here, we've got 1, 2, 3, 4, 5 copies of the sequence GATA for this particular VNTR locus. They're repeats. They're tandem repeats. That means the copies are next to each other. They're not scattered around the genome. They're variable number tandem repeats because the number of repeats in each allele differs. So these are different alleles of this particular VNTR locus. And the alleles are characterized by how many different copies of the repeat there is. This is allele 9. There are 9 copies of the repeat present at this position in the chromosome. So this is a position in the chromosome, VNTR locus D16S539, is a place on the chromosome where some people have 5 copies of GATA, some have 3, some have 6 or 7 or 9 or other numbers. So VNTR loci are so variable, they have so many different alleles, because DNA polymerase very often makes mistakes when it's replicating these sequences. And the mistakes that it makes aren't that it puts in the wrong base, but that it puts in the wrong number of copies of repeats. And that's illustrated in the next slide. So here's DNA polymerase replicating DNA that contains a repeat. And I've illustrated the newly synthesized DNA in green, and the template DNA repeats are shown in blue. Now, surprisingly often, DNA polymerase kind of loses its grip on the template strand. Normally, this isn't a problem because the sequences that it's replicating are able to base pair with the template strand. So when it comes back, because it hasn't gone far away, and when it comes back, it just zips right back to exactly the right place. But if the DNA contains repeats, short repeats like VNTR loci, it can come back at the wrong place in the string of repeats, as illustrated here. It should have come back so that this repeat here was base paired with this repeat here. But instead, it came back one repeat over. And the result is that although the template strand had 3 repeats in this segment, the newly synthesized strand has 4 repeats. When the cell divides, the cell is going to inherit this mismatched section of DNA, and if the repair machinery doesn't correct it, a daughter cell is now going to have an extra repeat in its array of repeats. VNTR markers occur at thousands of places around the chromosomes, but a particular subset of 13 have been chosen for legal and forensic work, and this is called the CODIS set of VNTR markers. There's actually another set used in some countries. CODIS stands for Combined DNA Index System. And these particular VNTR loci were chosen because they have the following properties. They're highly polymorphic, not just in Europeans, but in most populations around the world. they're on different chromosomes. As you'll learn in Module 7, this maximizes the chance that different children of the same parents will have different combinations. And they're typically repeats of just 4 nucleotides. This achieves the best balance between mutations that generate diversity and mutations that would change the alleles from a parent to a child, for instance, which would interfere with identification analyses. The frequency with which these markers change was addressed in one particular study. And what they found using the CODIS markers was that the frequency of changes in copy number was about 10 to the minus 3 per gamete per generation. So that's from parent to child. And this may seem like quite a small number. It's 1 in 1,000, but it's an enormous number compared to the rate of point mutations over the same period, which is about 10 to the minus 8. So this is 100,000 times faster mutation rate. Most of the mutations that occurred were a single repeat, a gain or a loss of one repeat, more commonly gains than losses. Longer alleles with more repeats, were more mutable because presumably they provide more opportunities for DNA polymerase to slip. And 4 times as many mutations occurred in the male parent as in the female parent. And this is consistent with what we know about the much larger number of DNA replications in the male germline. So here's a question. Think about a particular CODIS allele that was present in your grandfather. What's the probability that the allele that you inherited from your father, from your grandfather, has a different number of repeats because of mutation? And the answer is 0.002. 2 in 1,000 times you would have a different version, a different copy number. It's 1 in 1,000 is the mutation rate per generation, and there's 2 generations between your grandfather and you. Now, another advantage of using length differences as genetic markers is that they're much easier to detect than sequence differences. They can easily be detected by automated DNA analysis. Here's an example of a readout of the markers from one person. So here's the 2 alleles of one marker, 2 alleles of a different marker, a different one, a different one. In each case, they're shown as a trace, and the position of the peaks identifies the number of repeats. So at this locus, this person has a 7 repeat allele and a 9 repeat allele. At this locus, they have a 4 repeat allele and a 7 repeat allele. These analyses, these automated analyses, are very accurate and very reliable. The weak link in these studies is the humans in the study. So the chain of custody, as it's called, becomes vitally important. What happens to the sample after it's collected at the crime scene and then maybe handled by detectives, passed on into storage for some length of time, transferred to a laboratory for an analysis, generating data, which eventually makes its way to the courtroom? There's lots of potential here for human errors, and it's these human errors that make DNA fingerprinting not absolutely perfect. It's these human errors that give rise to the courtroom debates now on whether or not a particular bit of DNA evidence should be admitted into the evidence. So, we've finished our study of DNA fingerprinting by talking about what VNTR loci are, variable number tandem repeats, why they're highly variable in populations, how they have nevertheless stable inheritance, and the standard sets that have been developed for forensic work. And as I said, the big concern is not the DNA itself or the automated analysis, but human errors that can creep into the process. Coming up next, we're going to begin talking about real personal genomics. We'll talk about analyzing single genes in gene panels. I hope to see you there.